Today's video, we will discuss the polysaccharides. If we think back, monosaccharides means one sugar unit, and disaccharides means two sugar unit, and polysaccharides, poly means many. So, the first type of polysaccharides is starch. Starch is found in plant, and one of the major role is to store energy or energy storage. Now, there are two types of plant starch, the amylose and the alimopectin. Yes, amylose has a straight chain of glucose monomers. Now, it look like this. If would connect all the glucose monomers in a straight chain, now the bond that connects glucose monomers is an alpha 1,4 glycosidic bond. Now, I'm not gonna draw the whole structure, and let me show you why it's called an alpha 1,4 glycosidic bond. So this 1, 2, 3, 4, 1, 4. Now, let's be familiar with the alpha and the beta form. The alpha form, the OH group on the carbon 1 facing downwards, and the beta form of glucose has the OH group pointed upwards. So, this will have an alpha 1,4 glycosidic bond. Now, amylose has this type of glycosidic bond, the alpha 1,4 glycosidic bond. The alimopectin is different. It contains branches and structures. So it still has a straight chain, but occasionally you have chain that, so it still has a branch chain, so branches off. So it look like this. It look like a tree. Let's look at the structures. Again, I'm not gonna draw the whole structure, but I will show you why it look like. Now, from the main chain that will be explained of the amylose. Now let's talk about the bond of amylopectin. The straight chain contains alpha 1,4. This is 1 and 4. The alpha 1,4, same as the amylose. Now the branches of chain part of amylopectin has an alpha 1,6 glycosidic bond. So 1 to 6, yes, that's right. Now, alimopectin contains two types of glycosidic bond. The alpha 1,4 that's responsible for straight chain, and the alpha 1,6 that's responsible for branches that we see. Now, the next type of polysaccharides is glycogen. Glycogen is a polysaccharide made of glucose monomers. Now, starch found in plant, example is the potato, and glycogen found in animal, starch that found in animals. Glycogen is a highly branched like alimopectin. Glycogen has an alpha 1,4 and alpha 1,6 glycosidic bond with responsible for branch. Next, we have cellulose. It is a polysaccharides found in plants, and its role is not for energy storage, but it is a structural support, very long fiber in plants. This be- it has a beta bond branch going up between carbon 1 and carbon 4 of the glucose monomers. So cellulose link up the glucose unit by means of beta 1 4 glycosidic bonds. So 1 4 beta 1 4 glycosidic bonds. So that we to know regarding cellulose. Another polysaccharides is chitin. Chitin is similar to cellulose but found in insect that support to plant and has a structural functions as well and suppose as a energy storage. So now you familiar the basic polysaccharides or the basic units for polysaccharides.
In addition of figure 2.172, we are presented with another perspective of amylose, a type of starch molecules. From this angle, we can observe the linear structure of amylose, consisting of a chain of glucose units linked together by alpha 1 to 4 glycosidic bonds. This view highlights the helical nature of amylose, which give its characteristic physical properties and allows it to form complexes with various molecules, influencing its functionality in biological system and individual applications. This alternative view provides individual insight into the molecular architecture of amylose, contributing to our understanding of its behavior and potential uses.
Structural polysaccharides are carbohydrates that have a structural role. Plants have cellulose, which are polymers of repeated glucose units that are joined by beta linkages. Certain animals produce chitin that serves as a structural component, for example, of exoskeleton. Exoskeleton stiff covering of the outside of some creatures. Example of exoskeleton: beetle, earwig, shrimp, spider, and etc. Cellulose, which is a polymer of glucose with exclusive beta-1 4 linkages between the units, is an important structural component of plants and fungi cells. Cellulose with beta-1 4 links between glucose sugars. This is a beta-1 4 link. Notably, most non-ruminant animals are unable to digest this polymer as they lack the enzymes known as cellulase.
Good day everyone. Today I'm going to discuss about the ruminants in biochemistry along with the cellulose and the hemicellulose. Introduction. So, what is a ruminant? A ruminant is a mammal belonging to the taxonomic or suborder Ruminantia, characterized by a unique digestive system that enables them to efficiently digest plant-based food. Ruminants have a stomach divided into four compartments, which are the rumen, reticulum, the massa, and the ovimasum. Here are some examples of a ruminant animal. Rumen fermentation. So what is rumen fermentation? Rumen fermentation involves the breakdown of ingested plant material by a diverse community of microorganisms. These microorganisms includes the bacteria, protozoa, and fungi. These microorganisms ferment complex carbohydrates such as cellulose and hemicellulose into a simpler compound like volatile fatty acid, gases such as methane and carbon dioxide, and microbial protein. So these microorganisms are found inside a ruminant. So basically in their digestive system. So cellulose. What is a cellulose? Cellulose is a complex carbohydrate, also known as a polysaccharide. So we're talking about polysaccharide is a combination of monosaccharides. There's a lot of monosaccharides in the polysaccharides. Okay. That serve as the structural component in the cell wall of a plant. It is the most abundant organic compound on Earth and plays a crucial role providing strength and rigidity of the plant cell. Chemically, cellulose is composed of a long chain of glucose molecules. It is around, I think, 7,000 to 15,000 sugars linked together, which form fibers that are arranged in a parallel manner, creating a strong and resilient structure. Cellulose in a rumen fermentation. Primary substrate for microbial digestion. Cellulose is a complex carbohydrate found in the cell walls of plants, particularly in fibrous materials like grasses and forages that constitute the diet for ruminant animals. Enzymatic actions, where microbial population break down cellulose, producing volatile fatty acid. The breakdown of cellulose into glucose allows rumen microbes to ferment the sugars further. So, it is edible for humans. Well, in humans, cellulose acts as an insoluble fiber which cannot be absorbed, but partially broken down or fermented by beneficial bacteria of our large intestine. So how can we make it consumable? So the softening effect. Water molecules penetrate the plant cell wall, thus disrupting the hydrogen bonds. It is the water is boiled again. Actually, softening cellulose can be achieved through various methods such including cooking, soaking, and fermenting. Softening cellulose can enhance the palatability and the nutritional value of plant-based foods, making them more enjoyable and easier to incorporate into a balanced diet. Hemicellulose. Hemicellulose is a complex carbohydrate polymer found in the cell wall of the plants. Particularly in association with cellulose, it is a heterogeneous group of polysaccharides, meaning it consists of various sugar units such as xylose, glucose, mannose, and arabinose, among others. The difference between cellulose is a linear chain of glucose molecules, highly organized and rigid structure due to its linear arrangement of glucose molecules. Hemicellulose, heterogeneous group of polysaccharides composed of various sugar monomers, more shapeless and branch structure compared to cellulose. It is said that hemicellulose is only consists of 500 to 3,000 monomers of sugar, while the cellulose is around 7,000 to 15,000. That will be all. Thank you. Good everyone. In continuation with my previous report on ruminants, I'm going to talk about the silos in biochemistry. So we're going to talk about the structure, properties, sources, and its uses. First off is the structure. Xylose is a type of sugar known as a monosaccharide. Contains the simplest form of sugar with 5 carbon atoms. It is classified as an aldopentose, an aldehyde, functional group, and 5 carbon atoms in its structure. Its chemical formula is C5H10O5. Xylose exists in two forms. The D-xylose, which is the most common form found in nature, a specific spatial arrangement of its hydroxyl groups. L-xylose, which is a less common mirror image of D-xylose with slightly different properties. It has a 5-carbon backbone with a hydroxyl group OH attached to each carbon, except for the carbon at the end, which has a double-bonded oxygen atom. See double bond O forming the aldehyde group. Let's go with the properties. Its physical state is white crystalline solid. You can imagine it like a sugar. Melting point is 153 to 154 degrees. It is highly soluble in water, slightly soluble in hot alcohol. Taste slightly sweet, about 40% as sweet as the sucrose. Due to the presence of aldehyde group, xylose is classified as a reducing sugar. This means it can readily participate in chemical reactions involving the aldehyde group. The source. Xylose is primarily obtained from hemicellulose, a polysaccharide found in a plant cell walls. Hardwoods like birch and beech are good sources of xylose. So basically in trees. Xylose is included in the dietary carbohydrate. It is a part of sugar composition of fruits, cereals, bread, and vegetables like potato and peas and carrots. Its uses. Diabetic sweetener. Due to its lower sweetness compared to sucrose, xylose is a sugar substitute for people with diabetes. In dietary supplements, xylose is also sometimes used to promote beneficial gut bacteria. Medical diagnostic. A xylose tolerant test is a medical procedure that uses xylose to assess intestinal absorption. Thanks. Hello, good day everyone. In continuation in my last topic, which is the xylose, now I'm going to talk about the chitin in biochemistry. We're going to talk about its structure, function, and the conclusion. So first off is the structure. So chitin is a long chain polymer, meaning it is a large molecule made up of repeating subunits linked together. So these subunits are called N-acetylglucosamine sugars, which are structurally similar to glucose, but with an acetyl group attached. The GLC and acid units are linked by beta-1 for glycosidic bonds, forming a linear chain. Interestingly, chitin closely resembles cellulose, another abundant polysaccharide, except that the hydroxyl group in chitin is replaced with an acetylamine group. Well, as you can see on the picture, it is found in the second group, which is the NHCOCH3. Those are those is the acetylamine group. So cellulose is just in comparison, cellulose is a polymer of glucose, while chitin is a polymer of acetylglycamine. So there's just small differences in their structure. Just replace with the acetylglycamine polymer in a chitin side. Its functions. Chitin functions structural support. Chitin's primary function is to provide structure, support, and protection. It is the key component of the exoskeleton of arthropods such as insects, crustaceans, and arachnids, offering a rigid and lightweight external covering. Because cellulose is a hard in nature. Just that chitin is just a modification of that movement. In insect, chitin is crucial for muscle attachment. The striated muscles are connected into the inner surface of the exoskeleton, allowing for coordinated movement. Barrier function. The chitinous exoskeleton in arthropods acts as a barrier against environment, protecting them from dehydration, physical damage, and pathogens. Biodegradation. Chitin is biodegradable, meaning it can be broken down by enzymes producing by various organisms like bacteria and fungi. This degradation process plays a vital role in nutrient cycling by returning chitin back into the ecosystem. And there's a product that is actually very innovative. Like there's a chitin-based plastic which came from the animal what we call such as shrimp. It is a major contributor. Water treatment. Chitin represents an exciting development in water treatment technology. Its natural abundance, biodegradability, and effectiveness in removing contaminants make it a promising alternative for traditional methods. These are examples of chitin sources. Conclusion. Chitin's strong structure and diverse biochemical properties make it an essential biomolecule for many organisms. It provides structural support, facilitates movement, and offers protection. In nutrient cycling, chitin is a biodegradable biomolecule. Thank you and good day.
In this video, we're going to tackle about what is pectins, where to find, and its uses or importance. What is pectins? Pectin is a heteropolysaccharide, a structural acid contained in the primary lamella, in the middle lamella, in the cell walls of terrestrial plants. Where to find? Pectins found in fruits and vegetables, found in the cell wall of seed-bearing plants, present in the middle lamella and primary cell wall of higher plants, and pectins is abundantly present in the ripened citrus fruits like guavas, lemons, oranges, apples, and pears. Uses and importance of pectins. Pectin used as a gelling agent. It has the ability to form gel in nature, important constituent of jam and jelly. Pectin in the form of paste exerts a bacteriostatic activity and hence is used frequently in the treatment of ulcer and deep wounds. Pectin's consumption may result in reduced blood cholesterol levels due to its tendency to bind cholesterol. That's all for today's video. Thank you. In this video, I'm gonna tell you what is lectins, where to find, and how they work. What is lectins? It is a type of protein sometimes referred to as antinutrients because they can bind to carbohydrates and interfere with nutrients absorption. Where to find? Lectins were originally discovered in plants and animals. It is a protein in certain food that have two purposes. First, it acts as a survival mechanism to protect the plants against insects or animals. The second is, it helps the communication between cells. If a person is sensitive to lectins, it can cause an inflammatory condition in body system, including digestive tracts where food and nutrients being digested. Here are some examples of foods that high in lectins, such as beans, legumes, grains, and nightshades, or solanaceous crops such as tomato, potato, and eggplants. Animal products have lectins if that animals consumes food that high in lectins. Examples of animals are domestic animals such as chicken, ducks, hamster, rabbits, goat, and sheep. How does lectin work? Lectins are known as phytohemagglutinins. It will be obtained from the red kidney bean that binds to the membrane of T cells and stimulates metabolic activity, processes necessary for organism to remain alive. One lectins recognize hydrolytic enzymes. It is a protein in gut and work by breaking down other molecules into smaller fragments. Gut or gastrointestinal tract. It is a passageway of the digestive system containing mannose-6-phosphate. A major function is to bind and transport M6B enzymes to lysosomes. A mannose-binding lectins help defend against invading microbes. Some viruses and bacteria use lectins to recognize and bind specific carbohydrates residues on the surface of the target cells. Flu virus, for example, it carries a lectin known as hemagglutinins that binds to sialic acid and is essential for entrance of the virus into the target cells. After binding, the viral particle enters by endocytosis after the hemagglutinin has been separated by a protease, means proteins break into small pieces. After replication of the virus inside of the cell, hemagglutinin and the viral enzyme known as neuraminidase cluster in the cell membrane. Viral RNA and associated viral proteins cluster near this membrane site, and the new viruses bud off in a portion of the cell membrane after the hemagglutinin sialic acid linked to the infected cells is released by the neuraminidase, cutting the bond between the sialic acid and the rest of the cell surface carbohydrate. Drugs such as Tamiflu that interfere with neuraminidase works by preventing release of the viral particle. Unreleased particles will tend to aggregate and not function. Here are some potential benefits of lectins. While some lectins can cause digestive issues, research suggests that lectins may also have potential health benefits. Lectins may play a role in cell signaling and communication. It may have anti-cancer properties, and it may help regulate the immune system. Overall, lectins are a class of proteins responsible for several biological rules such as cell-to-cell interactions, signaling pathways, and several innate immune responses against pathogens.
I will be discussing about biofuels. Gasoline, diesel, natural gas, coal, and crude oil. These are the conventional fuels that we are using today as source of energy. These fuels are made by burning fossils which emit large amount of carbon dioxide into the atmosphere. This contributes to even worse climate change and other environmental problems. These energies are also non-renewable and finite, meaning once these resources are used up, they cannot be replenished right away because it takes millions of years. In this era of diminishing era of energy resources, We need to look for more sustainable alternatives such as biofuel. Biofuel is a renewable fuel made from the organic materials such as plants, algae, or animal waste, which can be used as an alternative to conventional fossil fuels like gasoline and diesel. So instead of burning fossils to produce fuels, biofuels make use of living matter like plants or animal waste and convert it to energy. This energy can be used as transportation fuel like what gasoline and diesel do. These are examples of biofuel products and the materials that they are made from. Also where these biofuels are commonly used for. So first, starting materials, plants. Example, corn, sugarcane, and soybeans. Biofuel product, ethanol and biodiesel. Common uses, ethanol, fuel additive, transportation fuel, and biodiesel is diesel engine vehicles and heating oil. Second starting materials is algae, biofuel product, algal biodiesel, and bioethanol. Common uses is algal biodiesel is transportation fuel, and bioethanol is fuel additive transportation fuel. And third starting material is animal waste. And biofuel product is biogas. And common uses is biogas is for electricity generation, heating, cooking, transportation fuel. So the fourth starting material is agricultural residues. And biofuel product is bioethanol and biogas. And common uses, bioethanol is fuel additive transportation fuel. And biogas is for electricity generation, heating, and cooking. Now, we already know that the biofuels are made from organic materials like plants, algae, and animal waste. But how is this made possible? Making these materials into liquid or gas fuels. First, feedstock preparation. Organic material is collected and prepared for processing, and it includes drying and grinding of organic material. Second, biomass conversion. For example, fermentation, used for producing ethanol from sugar or starch-rich feedstocks like corn or sugarcane. Enzymes are added and then fermented by yeast to produce ethanol. Another example of biomass conversion is transesterification is used for producing biodiesel from vegetable oils or animal fats. The feedstocks undergoes chemical reaction with alcohol, usually methanol or ethanol, and a catalyst. Third is refining and purification to remove impurities and ensure it meets quality standards. It may include distillation, filtration, and other separation techniques. And last is blending and distribution. The final biofuel product is often blended with conventional fuels. For example, ethanol blended with gasoline. And biofuels are distributed to fueling stations for use in vehicle, heating systems, or other applications. These are the advantages and disadvantages of biofuel. So, these are the advantages. First, renewable. Second, lower carbon emissions. Third, energy security. And fourth, economic development. And fifth, diversification of fuel resources. And last, potential for sustainable development. Meanwhile, this is the disadvantages of biofuel. First, land use computation. Second, technological limitations. Third, food security concern. Fourth, resource intensive. Fifth, life cycle emissions. And last, indirect land use change. To conclude my part, instead of heavily and solely relying on the conventional fuels, promoting biofuels during these times is crucial for addressing environmental challenges, enhancing energy security, and fostering sustainable development. And that's all. Thank you.
Heparin. What is heparin? Heparin is used to prevent or treat a certain blood vessels, heart, and lungs conditions. Heparin is also used to prevent blood clotting during open heart surgery, bypass surgery, kidney dialysis, and blood transfusion. Heparin and heparan sulfate are built of nonlinear change of repeating disaccharides unit consisting of a glucosamine and uronic acid. This is the structure of heparin. So glucosamine is an amino sugar and a prominent precursor in the biochemical synthesis of glycosylated proteins and lipids. Glucosamine is part of the structure of two polysaccharides, the tryptophan and the chitin. Glucosamine is one of the most abundant monosaccharides produced commercially by the hydrolysis and shellfish skeletons, or less commonly by fermentation of a grain such as corn or wheat. Glucosamine has many names depending on the country. Uronic acid the aldoronic acid are a class of sugar acids which both carbonyl and carboxylic acids functional group. They are sugar in the hydroxyl group farthest from the carbonyl group has been oxidized to a carboxylic acid. Usually the sugar is an aldose, but fructonic acid also occurs. The oxidation of the terminal aldehyde instead yields an aldonic acid, while the oxidation of both terminal hydroxyl group and the aldehyde yields an aldaric acid. So, fibrinogen. Fibrinogen is a glycoprotein complex produced in the liver that circulates in the blood of vertebrates. During tissues and vascular injury, it is converted enzymatically by thrombin to fibrin and then to fibrin-based blood clots. Fibrin clots function primarily to occlude blood vessels to stop bleeding. Fibrin also binds and reduces the activity of thrombin. The structure of thrombin. What is the effect of the fibrinogen? Fibrinogen is one of 13 coagulant factors responsible for normal blood clotting. When you started to bleed, your body initiates a process called the coagulation cascade or clotting cascade. This process causes coagulation factor to combine and produce a clot that stops the bleeding. So, anticoagulant. Anticoagulant are medicines that help prevent blood clots. They're given to people at a high risk of getting clots to reduce the exchange of developing serious conditions such as the stroke and heart attacks. A blood clot is a seal created by the blood to stop bleeding from wounds. Angiogenic is the psychological process through which new blood vessels from the pre-existing vessels form in the earlier stage of vascular genesis, and angiogenesis continues the growth of the vascular mainly by process of sprouting and splitting, but processes such as quiescent angiogenesis, vessels elongation, and vessels cope shown also by a row. So that's all. Thank you.
Good afternoon, everyone. Good afternoon, Sir Alistair. I am Janelle Kahas from BSA-1B. So the topic that I am going to discuss is synovial fluid. So, what is synovial fluid? Synovial fluid. It is a viscous, transparent fluids found in the cavities of synovial joints, providing lubrication and nourishment to the joint tissues. Among its key components is hyaluronic acid, a large molecule known for its lubricating properties. So, what causes synovial fluids build up? The causes of synovial fluids build up. In an active, healthy person, the most common causes of synovitis is overuse of the joint. For example, in athletes or people whose jobs involve repetitive stress, movement such as lifting or squatting.
So now let's forward to our next topic, which is function in skin. And now let me show you something. And now I'm going to share you of what are their function. There. And as Miss Kahis discussed, I'm going to show you also what is the synovial fluid. So this is the synovial fluid. This one. These are the So first is the extension muscle. So when we say extension muscle, any of the muscle that increase the angle between member of a limb, as by straightening the elbow or knee or bending the wrist and or spine backward. So the movement is usually backward. So like this. So second is the synovial cavity. So when we say synovial cavity, the synovial fluid inside your synovial membrane lubricate the part of your joint as they move. The fluid slide between pieces of bone or cartilage to help them move smoothly and without rubbing against each other. So the third one is the bursa. So when we say bursa, the main function of the bursa is to reduce friction between moving part in joint of the body. In addition, they act as a cushion that absorb pressure present in some joint. So the fourth one is the joint capsule. So when we say joint capsule, the joint capsule isolate and cover the entire joint. It consists of two layers, which is the fibrous layer, that is the outer layer, and the inner one is known as synovial membrane. So the fifth one is tendon. So when we say tendon are the soft fibrous tissues that connect muscle to bone. Their main function is to transfer muscle generated force to the bony skeleton, facilitating movement around a joint. So the next one is the enthesis. So when we say enthesis, an enthesis is the site of attachment of tendon, ligament, fascia, or capsule to bone. There are two types of enthesis, fibrous and fibrocartilaginous. Fibrous entheses are found at the attachment of tendon to the metaphysis or diaphysis and are composed of thin fibrous tissue. So the next one is the flexor muscle. So when we say flexor muscle, the flexor constructs to bend a limb at a joint. Then when the movement is completed, the flexor relax and the extensor construct to extend or straighten the limb at the same joint. So the next one is the epiphyseal. So when we say epiphyseal, tip of the long bone typically responsible for the articulation. The epiphysis is also the primary source of the red bone marrow in long bones. So the next one is the articular cartilage. So when we say articular cartilage, it is to allow motion of the musculoskeletal system to bear weight to hold the skeleton together. So the last one is the ligament. So when we say ligament, a ligament is a fibrous connective tissue that attach bone to bone and usually serve to hold structure together and keep them stable. And that's all. So now let's forward to the next topic.
Uronic acid is a usually hype ingredient, and for me, this is not an ingredient. To hold water more than 1,000 times molecular weight, creating gel substance, and it is sugar molecule, so effective when used in the dermis. Uronic acid plus 4,000 Dalton. The 500 Dalton rule means that larger molecules are unable to penetrate the epidermis, so HA is too big, so it must be it must be combined mineral oil, shea shea butter, and silicone. Uronic acid, also known as uronan or uronate, it's a glycosaminoglycan. So, pause muna tayo. Glycosaminoglycans or mucopolysaccharides are long linear polysaccharides consisting of repeating disaccharide unit. The repeating 2 sugar unit consists of a uronic sugar and an amino sugar. Except in the case of the sulfate, glycosaminoglycan keratan, where in place of the uronic sugar, there is a galactose unit. So, GAGs are found in vertebrate and bacteria. So why? So because of GAGs are highly polar molecules and attract water, the body uses them as lubricant or shock absorber. So there, so these are the structure of the glucosaminoglycan. So So this is the structure of the aminosaminoglycans. The repeating disaccharide unit GLCUA 1B to 3, GALNA 1B to 4 of chondroitin sulfate. For the polysaccharide nomenclature, see here R1, R2, R3 maybe have different values. So these are the structure of gluconic acid. UPAC name BD glucopyranuranic acid. Systematic UPAC name 2S, 3S, 4S, 5S, 4S, 5R, 6R to 3, 4, 5, 6 tetrahydroxyoxane 2 carbon carboxylic acid. Other name BD glucuronic acid GLCA. Chemical formula C6H10O7. Molar mass 194.139 G mole, negative 1. Melting melting point 159 to 161 Celsius, 318 to 322 to 432 to 434 K. And so this is the structure of the acetyl glucosamine. The UPAC name BD acetyl amino 2 deoxy glucopyranose. Other name acetyl D-glucosamine GLCNAC NAG. Chemical formula 8H15NO6. Molar mass 221.21. Melting point 211. So that's